Okay, hello folks. So this is another A-level, specifically AS level lesson in the DNA to protein series. So how the information in DNA becomes the proteins that make up you. And this time we're going to discuss the structures of ribonucleic acid. Okay, so RNA specifically. Now RNA is something maybe you got introduced to at GCSE. Certainly you would have been introduced to at A level, at AS level so far. Probably only the messenger type though, but it actually does a lot more than you think. And I'm going to give you some examples in this lesson of what else RNA does within the cell. Okay, so first things first, this is the sort of molecules that we're looking at. We can play a little spot the similarities, spot the differences. I hope you're pretty good at this already in terms of looking at structures. So things like amounts or presence of hydrogen bonding, numbers of strands, the structures of the sugars, so on and so forth. Because DNA you should know all about already. mRNA you've been introduced to already in this series, and the tRNA might be quite new. We mentioned it in the first video, but of course we're going to go into more detail with this now. Okay, so here's the spec, the idea of that what's a genome, right, and what's a proteome. Okay, what does that mean? Okay, what do these words mean? Genome is the entire thing, and then the structures of molecules, RNA, messenger RNA specifically, and transfer RNA as the two examples that are listed there on your syllabus. So quite a short bit of spec for this. So a really efficient way to do this is at the end of this lesson, if you are keeping notes on this, then obviously is to make yourself a table. Table comparisons are probably the best. If you get questions on this, they tend to be long answer. You know, 5 or 6 marks doing comparisons between two of these. Okay, so it's best to sort of have all your information nicely lined up. So I'd suggest if you wanted to sketch this out now, pause the video here, that's fine, or do so at the end. Okay, up to you. Right, so first thing, these two words, genomes and proteomes. Okay, so we've discussed what genes are, what chromosomes are, and that they code for protein. So the definition of a genome is the complete set of genes within a cell, and a proteome is the full range of proteins a cell is able to produce. Okay, now the study of both genomics and proteomics and their interaction obviously are related fields, but there's actually a hell of a lot that goes on between the genes and the proteins, and that's the sort of the topic of these few lessons. Okay, so genome is your complete set of genes, proteome is the full range of proteins. Okay, onwards. So hopefully you already know the basics, right? The basics of this, the basic structure of a nucleotide. There, look. Now this has been left deliberately ambiguous, right? This is obviously our base here, so our sugar here, our either ribose or deoxyribose sugar. We've got our phosphate group there, and we've got our base here. Now, in terms of bases, there are actually 5 of them, and they're split into 2 different types. So you've got the double-ringed ones there, which are A and G, adenine, guanine. We call those the purine bases. So this here is, of course, A, right? And then we've also got thymine, cytosine, and uracil, which are the pyrimidine bases. This is single-ringed structures. Okay. Now you should be able to work out and remember that obviously it's 5 bases there, so you're only going to get 4 at any given time. And it's actually DNA is AT, G, and C, and RNA is A, U, G, and C. So in RNA, the thymine here is replaced by the uracil, and you can see chemically why that's possible, as discussed in a previous video. Okay, that methyl group there. So, if you're asked the very sort of broad question to compare DNA and RNA, here's the kind of things that you might say. Do they both have phosphate groups? Well, of course, yes, they do. Okay. What is the pentose? And that's why I left it sort of slightly ambiguous on the last slide, was deoxyribose on DNA. So that carbon 2 has just 2 hydrogens on it, not an OH group. It's lost that oxygen. And ribose has got another OH group there, which makes it more reactive, right, less stable. Bases, as we've said, ACTG and ACUG. How many types of DNA are there? Well, really, DNA is DNA. I mean, we talked about how it's packaged and how it's structured and what its job does. Okay, but as far as the double helix goes, that's pretty much what you've got. Okay, in terms of the RNA, right? You've actually got three types, and I'm going to mention all three, but you need to know for the spec, as we've just seen, two types. Okay, are they both polymers? Well, yes. Are they both made of nucleotides? Yes, absolutely. In terms of size, DNA strands are much, much bigger than all the types of RNA. But notice I use the word bigger, and what's written there is the word smaller. These are comparative, okay? They're only big or small in comparison to each other, and you are required to compare these types of RNA to DNA. So smaller is the way forward. Okay, and you can see our little structural diagram here. Look, there's our difference in our OH and H group there, and there's our thymine and our uracil differences as well. Okay, so that's if you get the generic question of what's the difference between DNA and RNA. But let's now look at the specific types of RNA you might be asked about. So messenger RNA is the one we've already talked about and you may know something about. Okay, this is the one that carries information from the nucleus to the ribosome, right? And basically it's a complementary copy of the DNA. So it's made during transcription, right? That's the process there, right? It's made during transcription. And this is when a gene is switched on. When you want to use that gene, an RNA copy is made. And you can see the complementary base pairing forming that RNA there in transcription. Okay, more on transcription in detail in another lesson. So mRNA is still a nucleic acid and it's still RNA, right? But the specific things about it, of course, right, we've already said that this is a single-stranded molecule, not a double-stranded molecule. So there's your first difference between RNA and DNA. Now, we know this bit, it contains ribose instead of deoxyribose, and we know this bit, it contains uracil instead of thymine, okay, because it is, of course, RNA. The other bit specific to mRNA, it has no hydrogen bonding on its own. So when it's just out there on its own, the bases are not hydrogen bonded to anything. You'll see why that's important when we get onto the next type of RNA. Okay, so it's got no hydrogen bonding at all, okay? And there's that underlined important point again, it is much shorter than DNA. But of the RNAs, it's actually probably one of the longer versions. So it's a lot shorter than DNA, but actually longer than tRNA or the other type, which we're going to get to. Okay, that's why it's important that you have these comparative terms in. Very important indeed. Right, so moving on. Last point, and this is very important in terms of actually having this thing as a temporary molecule, flexible so cells can switch genes on and switch them off again, is it's got a much shorter life than DNA. It's a lot less stable, okay? Which is good, because of course if we started switching on a gene, made a load of RNA, and then it never went anywhere, then that protein would be being produced forever until the cell popped, or something like that, okay? So it's good that the RNA actually goes away. Right, so that's mRNA, and I'm assuming you know a bit about that, and then this might be the genuine new stuff, okay? So tRNA here, this thing, right, it's called transfer. That's what the T stands for, is transfer RNA. And there are lots of different types of tRNA. As we looked at when we looked at the genetic code, there are tRNAs that are what their job is an adapter molecule to bring amino acids into the ribosome during translation. And each tRNA is specific to one amino acid. Okay, so important bits. Look, you've got your 3' end, you've got your 5' end, just like any other strand of nucleic acid. Okay, and then we've got this thing called an amino acid attachment site, and on the other end, an anticodon. As you can see, this is folded over. So each tRNA has got this thing on it called an anticodon. Now that's not your mum's northern sister called codon. Okay, for the accent fans out there. It basically means the opposite to a codon, okay? They're complementary to the codons on the RNA molecule, right? So these literally act as adapter molecules. So to give you an example, let's say the RNA had AUG, which is the start codon. It's the one that tells the ribosome, right, start here, this is now going to be a protein. Right, that AUG, right, the anticodon, the bases down here, right, on the tRNA would be UAC, okay? So that they could hydrogen bond together and drag the correct amino acid into the ribosome, okay? So that is how they're adapted. Now there are a series of special enzymes that are there to make sure that the right amino acid is placed onto the right tRNA within the cell. But what you end up with is you end up with a very effective little tow truck that on one end can interpret the language of nucleic acids of RNA, and on the other end has got a protein to form a polypeptide. So this is involved in the process of translation, translating between the language of nucleic acids and the language of protein. Okay? So other things that you should know, which could come up in a question, it is single-stranded, but it does have hydrogen bonds. And this is how they catch you out, okay, compared to RNA. So RNA is single-stranded, but doesn't have any hydrogen bonds. It's a linear, open-form molecule. Because what's going to happen? It's going to be read by a ribosome, okay? tRNA is still single-stranded, but it does have hydrogen bonds because it's folded up on itself, okay? So if you were comparing this with DNA to tRNA, you would say, well, they both have hydrogen bonds between bases, okay, which RNA doesn't. So be careful that you've got the difference between whether or not it's single or double-stranded and whether or not there are hydrogen bonds, okay? It's shorter than either DNA or mRNA, okay? It's very, very short. But again, use the comparative terms, use the on the end. And we sometimes describe its shape as like a clover leaf shape with these 4 loops, well, there's 4 ends, 3 loops, and then this one here in the middle loop having the anticodon. And I always say, and I put this in quotes because I don't want you to write it in an exam, I always think of them as little tow trucks. Okay? They drag in the right amino acid into translation, and the ribosome then knows which amino acids to put together to make a protein. Okay? So that's tRNA versus RNA. Now, it is entirely reasonable for an examiner to ask you to do something like this. Know your way up and down the code between DNA, mRNA, and tRNA. So it's entirely reasonable for them to give you the code on any of those things, the DNA, the mRNA, or the tRNA, and ask you to work out what the code on all the others would be. So what I suggest you do now is pause the video and have a little look, see if you can work out for each of these what the other 2 is going to be. Okay, so pause the video now and we'll get the answers shortly. Okay, hopefully you've done that. So we'll start down here, look, GAT, well, G is complementary to C and T is complementary to A. Now if this was DNA, then A would be complementary to T, but it's not. RNA, so it's U, CUA. So GAT goes to CUA, okay? And then in the tRNA anticodon, the middle loop of the anticodon would stick to that, that would make GAU. Okay, so GAU would stick to CUA. Okay, hopefully you got that. Same with this one working backwards, CUG, okay, well C goes to G, U goes to A, and G goes to C. Okay, that's what they would be complementary to. And then working forwards, well, it's going to be the same, it's going to be GA, but in this case now it's going to be C and not U. Okay? And then working all the way backwards from the tRNA anticodon, AUC going backwards would be UAG. Okay? Be UAG, and going backwards from UAG would be ATC. Okay? Hopefully it's clear how we got to that, but you can work out how that works. Right? And obviously if you can't, you need to go away and practice that because it is easy marks on an exam. Okay, so those are your main 2 that you have to know about. But there is another one, right, which is called ribosomal RNA, which is one of what we call the functional RNAs, an RNA that does something. Okay? So ribosomes, now I've put a picture of that unfortunately, but ribosomes are made of a combination of RNA and protein. Okay, so they're made of protein as well. Obviously in my excitement of having a little spinning animation there, I've put it over the words and protein, but they're made of a mixture of the 2. So for those of you who've looked into this, well, you all should know by now that ribosomes, look, have a large subunit and a small subunit. They're like a burger bun, and they sit round the RNA and they run along it. It would be natural to assume they're made of protein. Actually, they're made of RNA. And the more primitive the organism you look at, the more it looks like they're made of more and more and more RNA and less and less and less protein, suggesting the protein actually came in later, okay? And that initially, these functional RNAs had lots and lots of roles in the cell, particularly in the early part of our evolution. So this here is one example. This is the small subunit. So this is that bit there. Right, this is the small subunit of ribosomal RNA in this thing called Thermophilus, which is a bacteria that can live in hot springs. Very well studied, very well understood, used in a lot of biotechnology applications. Now, for reference, the ribosomal RNA is the orange part, so all that is orange, and the ribosomal protein is blue. So you can see it's quite a high percentage of that functional molecule that's actually RNA doing something. Not just RNA coding for something or translating for something, it's actually doing something. Okay? Now that obviously is not on the AS level spec, right? But it is the reality, it exists. And also, the more we look, the more we seem to find in nature RNAs that actually do stuff. Okay? So this thing here is actually part of an RNA enzyme. An enzyme made with an active site and tertiary structure, but not made of protein, but made directly from the RNA. Okay? And they've been sort of named ribozyme, right? They've been named ribozyme, so these RNA enzymes. Turns out they're very important in cutting, splicing, modifying, changing other RNAs and other nucleic acids. And this particular one here is a motif called hammerhead, which I quite liked. Right? Hammerhead ribosome, which is involved in cutting over RNA. So basically going over RNA and just cutting it. Okay? So we'll get back to splicing and RNA cutting in another video, but those are our types of RNA. Okay, so we've got DNA, we should know all about, mRNA, tRNA, and then slight extension, rRNA as well. So now you need to make sure that you finish your comparison tables off, okay, and do some top tips here. Okay, so for when you get questions like this on exams, here are your top tips. Look very carefully at what type of molecule is being asked for, right? Are you being asked to compare DNA to tRNA, tRNA to mRNA, mRNA to DNA, or just the blanket RNA to DNA? Okay, what are you actually being asked to do? Check the questions very, very carefully, right? Because obviously you might end up with reading one question and answering another. Okay, use the suffix. I would say very much. Okay, use the suffix. Really important. Shorter, longer, not just short, long. Because again, it goes back to checking the questions carefully. Okay, and make these comparative statements. Right. So be careful that you're answering. Really particularly carefully depending on what the questions are asking for. So the hydrogen bonding thing is a classic. Whether it's single-stranded with hydrogen bonds, single-stranded without hydrogen bonds, or double-stranded with hydrogen bonds, be careful what you're looking at and what you're answering. Okay, make sure they're comparative because that's usually what the questions are asking you to do. So that's your types of RNA. I hope that's nice and clear for you, and we'll see you next time when we're going to go into more detail on how transcription and translation work in taking that information from the DNA to protein. See you then.